Well, good afternoon. I can see there's still attendees joining, so we'll wait a little bit.
Counting up. Yeah, good evening from Denmark. Where it's 3:30. We're still counting up now. I think the count has kind of stopped. So I would like to say hello and welcome. My name is Jonas. I am the head of the scientific support here in Chemometic in Denmark. And I'm happy to see that a lot of people signed up, more than 400 signed up for this webinar. And I'm happy to be joined by Annemieke as well today. So Annemieke did her PhD at the University of Edinburgh and her postdoctoral work afterwards on programmed cell death, so apoptosis in neutrophils connected to inflammatory diseases. And she will be talking to us about how to deal with issues related to aggregated cells. And then after that, we will take some questions. So about half of this webinar will contain Annemiekes presentation, and then the other half will be replies, hopefully, to you or to your questions that you should just add into the question section. So without anything further, I will ask Annemieke if she could start the webinar.
Okay, let me just share my screen. Hold on. Right, there we go. Well, thanks everyone for attending. This is really just meant to be a very short presentation, and we really hope that in the Q&A session we can address your specific counting issues. So if we go to this slide. So the key takeaways is in this webinar, we want to talk about cellular aggregation, how to break apart aggregates. There's various methods that we've all probably tried to use in the years in the lab, and some of the practical laboratory solutions as we see it for disaggregating cells. And then of course, we've got our Q&A. So Chemometic was founded in 1997. It's now a global provider of cell counters. But when they started in 1997, it was actually with the aim of producing an instrument for dairy farmers to be able to count somatic cells in milk. And so therefore the instrument had to be really simple to use and portable as well. Obviously, a farmer does not have a laboratory and pipettes at his or her disposal. So yeah, so that's why it was designed with such simplicity. And really that simplicity still exists today. And even though we do have lab and pipettes at our disposal, I'm sure we all really prefer that it's simple to use as well. So what is cellular aggregation and why does it matter to me? Well, aggregates are a problem, and in terms of cell counting, it's very, very difficult to count or enumerate an aggregated cell population. And our definition of cellular aggregation is when the percent of cells in aggregates is of 5 or more. And cell clustered together are not easily dissociated. So some examples of cells with a tendency to aggregate are stem cells, iPSC cells, epithelial cells, cells grown in microcarriers. These are all, and these can be really tricky to disassociate and enumerate. So I just wanted to show you an example of a nice single cell suspension. And these are Jurkat cells, really easy to count, no aggregation problems whatsoever. But then when it comes to other cell types, so if you keep this image in mind, you come to other cell types, you can immediately see from the image that there's big clumps of cells, and these are MCF7 cells. And then we have another example of CHO cells here. And with these kind of clumps, it would be very hard to get an accurate cell count out of that. And here's just a reflection of why aggregates are a problem. So whether it be machine or human cytometer, it's very hard to enumerate that. So if you look at first glance, you count them, you get 1, 2, 3, 4, 5, 8 cells. However, when we deaggregated them, there were 11 nuclei. So, and that's because when you go in that kind of 3D cluster of cells, you don't know what's lurking within that, how many other cells are lurking within that cluster or underneath that cluster that you can't see. So how can we break down aggregates? So the typical methods are really chemical and enzymatic lysis, add a chelator, proteolytic enzyme, or you could do mechanical, mix lightly with a pipette or mix harshly with a vortex. And also addition of DNAs to help you break down these aggregates with gentle mixing. So these are all sort of the things that are routinely done to try and deaggregate our sample. This was, and just to sort of look at the complexity of how you can test and the sort of experiments you need to go through to find a method that will deaggregate your particular cells. So this was a paper by Jaeger et al., and it was to disaggregate neural progenitor cells. And you can see that it won't be very easy to read all the different experiments, but they were basically trying different combinations of EE, acutase, trypsin, EDTA, using a P200 pipette or a P1000, and for how long. And so it was a huge amount of work to find out. I think in the end, they found that the best method was 10 minutes of incubation with EEE, and then 10 minutes of incubation with trypsin and EDTA. So that's a 20-minute incubation time for doing a disaggregation. And so it's incredibly time consuming, and you're also adding in quite a lot of variability, especially when it comes to viability, because over trypsinization of your cells is obviously not a good thing. It can release DNA into the media. Then you have to add DNase 1 to dissolve that, because free floating DNA will further cause aggregation by acting like a glue. So it's also not it's quite a complicated process going down that road. So here at Chemometic, we developed the method by, if you could lyse your cells, would that be easier? So lyse your cells, then stain your cells with nuclei or DAPI, quantify total nuclei under a microscope. So your nuclei are acting for you to quantify total cells per mil. And then for your viability, you could not lyse your cells, but stain cells with DAPI or PI. And quantify the dead cells under the microscope, because the DAPI and PI will only stain the nuclei. This part of the viability is based on the assumption that anything dead in the media will be free floating, or if it is stuck in a 3D cluster cells, a dead cell, then you'll be able to discern where the nucleus starts and ends. It won't be all absolutely glued together like if it were a whole cell. So here, just a basic sort of overview of the options for when you're coming to try and deaggregate your cells to get a decent count. So you can do the mechanical separation, trypsin, collagenase, etc., and then add DNase 1 to mop up any free-floating DNA, or you can use the lysis buffer, which will remove nearly all aggregates. So how do we do this? So here's an example of a tube with heavily aggregated cells in it. You do your first count with one of our proprietary cassettes, and then you count nuclei. So you lyse the cells and count nuclei to get your total cells per mil, and then you go and interrogate the sample again. Again with the without lysing, and look at the staining for the dead cells and get your viability that way. This is the count and viability assay. So these are nuclei just stained, evenly dispersed. And here's really an example where what we have done is taken freestyle CHOs And then you can see on the right, the real heavy aggregation. And then on the left, the lyse sample and only the total nuclear count. And it's quite apparent that, again, whether using a microscope or a cell counter, that the lyse sample is just going to give you a more accurate count. And to illustrate this, we did this in the lab with lysis method versus a non-lysis method. And there was an average of about 48.8% aggregation in this MCF7 cell line. And if you look at the bottom of the figures, the total cells per mil, you look at the repeatability of it, and then you look at the average. So the average, both methods gave an average of about 2 million cells per mil. But if you look at the CV, in the non-lysis method, you're just going to get an incredibly high CV, 17.34% versus 2% using the lysis method. And the bar chart is just to reflect that huge difference in CV that you get with if you don't lyse your cells. So that is just the end of methods for dealing with aggregated cells. I'm going to now stop sharing my screen, and if anybody has questions, can you please put it in the Q&A box, and I will read it. So, but we actually had 2 questions prior that was emailed prior yesterday, so I'm going to read them out to Jonas. So Jonas, how do you know the aggregates are a property of the cultivation conditions and are not an artifact of the measurement system?
So thank you very much, Annemieke, for that very clear presentation. So how do I know that it's actual aggregates and not an artifact from our measurements?
Yes.
Usually what you would see in case you have a heavily aggregating cell culture is that you will have the ability to see it with the naked eye. Sometimes you would like to have a microscope inspection available, so that you can see whether you already beforehand did dissociate the cells correctly, if it is your aim to have single cells. So in terms of whether the system that we're using is creating aggregates, we don't see that with the single cells, as you also showed. The Jurkat cells are in themselves nicely isolated as single cells. Usually it is recommended to have your cells in the full medium, because if you use other kinds of media, then you might see some electrostatic changes in the surface of the cells that may cause increased aggregation. So of course, there's a lot about sample handling, and often if you want to have the best quality of the sample for a fresh cell count, then you would like to have your cells in the full medium and count them in the full medium. So I think that taking the cell suspension up in full medium in the cassette, which is one of the instruments that we use the most, is a good representation of the cell culture as such.
Right, okay. And then further, they ask, are there significant shear forces in the nucleocassette that causes cell disaggregation, thereby creating a false low aggregation value?
Okay, that's an interesting question, because what we are looking at is mostly to avoid aggregation, so that we can have the single cells. But apparently the person asking this question is interested in having the intact, looking at the intact nuclei or aggregates. Anyway, I would say that yes, probably there will be shear forces, or you will have shear forces, as you will have in a pipette in in any system. So in this case, the aim would be to have as consistent a shear force as possible. So if you want to compare different samples with different or with the same level of aggregation, or you want to have a cell sample that is as intact as possible between the samples, then you want to have the same amount of pipetting beforehand. So the same procedure leading up to the measurement of the sample. So you apply the same shear force every time, because shear force when dragging a cell suspension out of its environment is impossible to avoid. So in this case, yes, we do introduce a shear force, but we introduce the same shear force every time, at least if we are using the cassette. Actually, I have a cassette. I don't know if everybody is aware of the system. I just brought a cassette for. So the shear force would be when you take up the sample here. And this is how you take up the sample in the cassette, and then the instrument will put the sample into the measurement window right here where the cells will be measured.
So I've got quite a few questions coming up. So what is the biochemical basis of the lysis assay?
So we use two methods to isolate the nucleus. So the entire idea is to have the intact nuclei. So we need to apply something that removes the plasma membrane and does not remove the nuclei or messes with the nuclear membrane. And what we've found is that we have having a good combination of a low pH and adding either a non-ionic detergent or a cationic quaternary ammonium will remove the plasma membrane to an extent where we have in combination with a low pH, we have an intact fixed nuclei that we can easily measure. So we've seen that if you have too high a pH, you will have electrostatic surface of the nuclei that will cause the nuclei to aggregate as well. So it's really a fine balance. And again, if you have too low a pH, then you will cause hydrolysis of the DNA and it will diffuse out of the nuclei. So it's really a fine balance there to get the right amount in order to have the intact nuclei that can be counted and separated.
Okay, so I've got another one actually that we often talk about. Lysis method is great, but it is done on a sample. However, with aggregates, it's hard to have a representative sample. Any idea for better sampling?
Yeah, that's tricky. So often we see that the biggest problem with aggregated cells is that taking out a representative sample. So one way of doing it is to take a very large sample out and add the lysis buffer. Then you will have, that is the best way to get a representative sample, to take out as bulk as possible, a sample, and lyse everything. Elsewise, I would do mild trituration. You can use a cell strainer as well. So obviously now we are having multiple different audiences. Some would be interested in just counting single cells from CHO cells or HEC293 cells that tend to aggregate and really don't like the aggregation. Other people are actually working towards making aggregates. So they're making clean meats, are making organ regeneration. So they would like to have as much aggregation as possible. And then you need to take out samples from this. And obviously in this case, a mild trituration or a cell strainer could come into play in order to dissociate the cells to an extent where you can do a good sampling. Once you have a good sample of your total cell culture, then you can lyse the cells and get a confident total cell count.
Okay.
I hope that answered the question.
Okay, and then we got, hello, thank you for a brief and clear presentation. I have one general question. In your experience, what is a better indicator for cell viability count? Count of live or dead cells? Which is more trustworthy?
So the most trustworthy is to count as many cells as possible. So whatever gives you the bigger count number will probably be the most trustworthy method. In this case, we're using a method where we do a total count on the lyse cells, so the cells that we are actively killing in order to get the total count, because as Annemieke showed you, we had this 17% CV on the aggregated cells, and then on the side we count the total amount of dead cells and make that relative to our total count. Because in this case, this is empirically what we found is the best way to get around this problem. But of course, in some cases, it really, if you have a very small number of dead cells to deal with, then you will get very high CVs, and that's often the problem. If you have a cell culture of 100,000 cells and you only have 1% dead cells, then you will have a very low N for dead cells, and you might in some cases not have any dead cells in your window. So
okay, and then we have another one. Do all chemometic cell counters use lysis of the cells?
no. So we have cell counters that uses the lysis as a solution for the total cell count, like the NC100, the SP100, and the YC100, and then we have the NC200 and the NC202, where you have a dual staining, so you have both Acridine orange and Derby to count in one cassette, both the live and dead cells. So but we grew out from a nucleo counting technology. So our base method is actually this lysis method that we've then further evolved into this assay to count aggregated cells.
Yes. And so also another question is how do you know if you're got too much variability? You know, what is an acceptable CV in counting?
For the total cell count, or for the, are we talking?
I imagine total cell count.
And again, it's all about the numbers. So if you have a very low number of total cells, then you would expect to have, just from the normal distribution of the cells, to have high CV. Usually we say that with 1 million cells per mil, you want to have a CV that's below 5%.
Yeah, okay. I've got one here. I'm trying to find it again. Where is it? Sorry. Yeah, we are using Accumax to remove aggregation. However, this product seems to introduce aggregates when comparing using the microscope, the cells in the aggregates turn blue after trypan blue addition. Do you have a suggestion what might be the cause? Well, I think it might be a bit what the paper from Jaeger was saying about, it might be over-trypsinization, and then you get release of DNA, which actually makes aggregation worse.
And we'll have both the DNA and the protein released, and that will stain for the trypan blue, so you will get these blue aggregates.
Yeah.
Makes sense. And
then
could be a good application for lysis solution.
And then also, is there any cells that you couldn't use this lysis method on?
Not in our hands. So we've had some challenges with the cells, like primary cardiomyocytes, where we see muscle cells that are effectively a syncytium. Then you can say, so if you do not have an intact nuclear membrane, then when you open the cell, the DNA will be released. So if you have cells that are stuck in a mitotic phase where the nuclear membrane has been broken down, then you will lose the DNA. So you can say that it is a requirement that you have an intact nuclear membrane. That's the only thing I can think of. We did solve the problem with the cardiomyocyte. We have a higher pH lysis buffer that we use used for that specific situation.
And then here, another one we've got, how to deal with yeast cell aggregates that disturb measuring the true growth rate from the sample from bioreactor. Aggregates disturb the optical density method a lot.
So we've developed a buffer for yeast cells specifically that we use in this case.
Okay. And then this is quite a long one. So as I understand, the cell viability is calculated from the lysed sample and the number of dead cells. But in one sample, the cells are disaggregated and the other not. This introduces some bias. Wouldn't it be better to determine viability using two measurements with non-disaggregated cells and a third measurement using the lysed cells?
That's an interesting suggestion. So two measurements
Wouldn't it be better to determine viability using two measurements with non-disaggregated cells and a third measurement using the lysed cell?
So at the moment, we actually have multiple methods for for measuring the viability. As we see that if you have extremely large aggregates or working with microcarriers, you might have very few dead cells in suspension, and that will cause a low viability measurement. But since we have in one measurement have both the viable cells and the dead cells, we can measure the viability from one sample, and then from the other sample we get the total cell count. I'm not sure introducing an additional measurement will create further certainty, especially because you might also introduce bias the more measurements you make. Okay. So, yeah.
And I think this one must have been from one of our friends. Which is the best cell counting system you got? Is it GMP compliant?
that would be the NC202 at the moment. If you're looking for a GMP compliant cell counter, that would be your go-to instrument.
So for suspension cell line and aggregate cases, is it suitable?
For
the lysis for suspension cell line and aggregate cell lines is, I suppose this person means the lysis method. Is it suitable?
It is indeed. But for many suspensions, so as a rule of thumb, we say if it's aggregating above 20%, then you should probably use a lysis buffer. But that's a rule of thumb. It depends on the cell line that you're using, that you're working on. It depends on how the aggregates look. As you mentioned in the beginning of the talk, we have a threshold that says aggregate is when you have 5 or more cells together, according to our algorithms. So you might have aggregates that are few but extremely large, or you may have multiple small aggregates. And of course, what you want to see is that, or the larger the aggregates are in themselves, the bigger is the need for a disaggregation reagent. The more complex is the algorithm that's required to separate these cells. So
I think we're out of time now. Actually, we're 1 minute over. So if anyone who's put a question in the Q&A, if we didn't get to your question, we will reply in the next couple of days by email. So thank you very much, everyone. Hope you enjoyed it and have a good day.
Thank you. Looking forward to answering your questions.